So welcome everyone. I would like to take just a moment to give a nod to all of the folks who have really helped with getting this virtual workshop series together. We've got a pretty large team that's putting things together. And so all of that information, of course, can be found in the bios that are located in the Google Drive. But on the Wisconsin Sea Grant side, we have Ann Mosier, who you've just been hearing from. And then Ginny Carlton has also been a huge part of getting this workshop series together. And Brenna D. Namer has also been helping us as well. She's actually the one that you've probably heard from with regards to getting information together for the bio. And then we have Cole Fisher. She's the program assistant who is helping Minnesota Sea Grant and the rest of us working on stuff. And we have Cindy Hagley, who recently retired, but is now volunteering her time to continue supporting really great workshops like this. And then we have Lori Hack. So Lori Hack is a high school teacher, and she has also played an instrumental part in getting this workshop together. As a teacher, she's been the one that we've sort of used as a sounding board to say, hey, Lori, does this seem like, you know, a pretty good way to go about things? And so she's been advising us and assisting us in a myriad of ways to put together a good workshop. We also acknowledge that I'm sure all of you will also like to introduce yourselves. Given the size of this group, we will be doing that during our joyful hours, and then probably a little bit in our breakout sessions here during the workshop, just in case you are not being part of the joyful hours today. And the last thing that I want to introduce or just mention is that Wisconsin Sea Grant, Minnesota Sea Grant, and the other Great Lakes Sea Grant programs are all part of an organization that's called the Center for Great Lakes Literacy. So this center is made up of Great Lakes Sea Grant educators, teachers, and scientists from all around the Great Lakes that work together to improve Great Lakes science and education and literacy. And so you can learn more about the Center for Great Lakes Literacy at seagull.org. And so that's a little bit about us. Most of the funding that comes for this is from the Great Lakes Restoration Initiative, and that is the funding source that helps keep the Center for Great Lakes Literacy rolling. And with that, Anne, I think that we were going to roll into doing a couple of polling pieces, and so I think we could probably get those rolling right now. We've got a couple of questions to ask you. And so this is your first time to work on being a bit interactive. The question, of course, is how many Laurentian Great Lakes have you visited? And so we've got several responses rolling in. People are clearly finding the way to respond here. We've got about half of the respondents now. We'll give you guys a couple more seconds. Some folks may have called in on the phone, and if you have called in on the phone, you probably do not have access to the polling. And it looks like we may be topping out at 27. So, okay, so how many Great Lakes have you visited? There are 0 people in this group that have visited 0 lakes. So everyone has visited at least 1 lake. And we have 3 of you who have visited all 5 Great Lakes. So thanks for that.
Marty, I'll just add in that we have Sue from Superior has visited 2, and Celine also has visited 1. Just wanted to add that in.
Okay, thanks. All right, and I think here I can share results. Someone is enjoying a really crunchy snack, and I'm glad that you are, but if you can find your mute button, that would be helpful. Thank you. Okay, and then I think that's it. So was that our last poll, Ann, or did we have 2?
We have a poll later on in the workshop.
Okay. That's right. So I will get out of that and continue on with our introduction. So another couple pieces of housekeeping that we'll cover before I turn it over to our guest speaker today is that you should have received a document in an email sent out by Cole Fisher via the Center for Great Lakes Literacy email for an action plan. Now, this action plan is a document that you can keep as a reference for now, but be thinking about filling it out. We recognize that You may be interested in a lot of the content and topics that we are covering, but that there may not be a way or desire to incorporate all of that into next year's lessons. We recognize that next year is a particularly challenging and interesting time for teachers, but we do want you to be familiar with this document and be thinking about the things you may want to plug into it. This will help us in the future when we reach back out to you. It's a document that then we can both share and we can remember sort of what our thoughts were during the virtual workshop series, and also see how things are coming along throughout the school year. So it just helps serve as a guide. We will be looking to turn it in at the end of the workshop series. So just the last couple of things, remember that the session is recorded and we're also recording the chat. And then we will be able to do some question and answer session as well. And there may be just a slight change to the schedule that was sent out. The timing has needed to be refined and tuned as we've gone along. And so it should follow pretty much as expected, but it could be a slightly bit different. And that's pretty much it. Right now, I would like to introduce our scientist of the day, Dr. Valerie Brady. Dr. Brady is the Associate Director of Research for Minnesota Sea Grant, and she is also a Senior Research Program Manager at the University of Minnesota's Natural Resource Research Institute. And we also have Holly Wellard-Kelly. She's a research scientist and a taxonomy specialist, also with the Natural Resources Research Institute. And with that, Val, I would like to turn it over to you.
Remember to unmute. And I am having a little bit of jitter on my internet, so I am going to beg all of you to please turn off your video and give me as much bandwidth as you can. And I'm going to share my screen with you. All right. Marty, can you confirm that I am showing the, and I'm getting feedback. Can you confirm that I'm showing the presenter view? And I can't hear you.
You are sharing the presenter view.
Wait, the correct view or the wrong view?
The correct view.
Great, thank you. Okay, so today we're going to talk about one of my all-time favorite topics. These are the aquatic invertebrates. So in my job, I use aquatic invertebrates and fish to tell the condition, the health of lakes, streams, and wetlands. And Marty and Cole and the team have put together a cool activity how you guys can do this with your students. I started playing with bugs when I was a little kid. I think I had my first butterfly collection when I was like 7. Played in mud puddles, finally figured out I could do that for a living. Took me through undergrad to figure that out into grad school. And I'm not going to say I get rich at this, but I get to be outside playing with really fun bugs, doing stuff I love, and hopefully helping save our aquatic resources or make them better. So that's a great job to have. I also want to say, please, please, please encourage your girls in science. I am happy to provide you with pictures of girls doing science to inspire young women to stay in science, especially women of color. We've employed lots of women on our team over the years. They do great. They have the muscle and the strength and the stamina to handle it just as well as any of the guys. They love the bugs and the fish. It's a fun time and it's great. It's a great job, and we need more girls and people of color in science. So please do what you can to encourage them. I can provide you with pictures if you need to show them girls in action. All right, macroinvertebrates. So funny name because they don't look macro, although everything's relative, right? It means you can actually see them kind of without a microscope, right? As in not completely tiny. And then of course, invertebrate. They don't have backbones. They're not like the mammals. So they're really familiar creatures in our everyday lives. You all know them, all the things we call bugs. And I'm going to use the word bug as a slang term interchangeably with the word macroinvertebrate because macroinvertebrate's a lot longer to say. So we're going to talk about the aquatic ones. So that just means they live in the water, right? They live at the bottom of streams. They swim around in rivers and especially swim around in lakes because they're little tiny, so they're not that good at swimming. They're really important parts of the food web, but they're just so cool on their own, and they have their own little food web thing going on too, and I'll talk about that a little bit. So I've been told I don't have time for a whole lot of anecdotes, but just this guy right here, this is a helicocycid caddisfly. He's the size of my little fingernail, and he builds a little case out of sand grains spun together with silk that he spins in his mouth parts, and if you flipped it over, it would look just like a snail shell. People see these on rocks and they think they're snails. They're really cool. And I'll talk about other cool things as we go along. So why are they important though? Besides the fish eating them, which my husband's a fisheries ecologist, so he's like, yeah, you do the fish food. Well, they're more than that. They are important fish food, but the fish can swim away if something bad happens, right? Fish are pretty good at swimming. This little snail over here, something bad happens, he's not going very far very fast. So if something really bad happens and it kills him, it's going to take them a while to crawl back, right, and recolonize that area. So we're going to see that signal in the stream or the lake or whatever for a while to come. The fish, you know, if the bad thing goes away, fish may come back. And if you sample the fish, you might not know that, hey, 2 weeks ago, something bad happened here. But if you don't find any of the invertebrates, because they're all dead and they haven't come back yet, that's a clue. So, I mean, that's an extreme example. It's usually much more subtle than that. Some are there and some aren't there, and I'll talk about that. But that's one of the ways that, and one of the reasons we use macroinvertebrates. They have faster life cycles than fish, and they can't escape the way fish can. The other thing is people say, well, why don't you just test the water chemistry? Well, somebody spills a nasty chemical in a stream, it's going to wash downstream pretty fast. Now, it may wash downstream, killing all the invertebrates as it goes, but if you're not there right when it happens and the water washes downstream, you weren't there to take a sample, you come back a few days later, you won't know by testing the water, but you can still ask the bugs what happened. All right, so the trick is we have to be sleuths. And this is where you can get, I hope, get your kids excited about this. We have to be sleuths to understand what the invertebrates are trying to tell us about where they live. Because we all know that different critters have different kinds of needs. And with the invertebrates, some of them like to live in streams, some of them like to live in lakes, some of them want to live in rocks, some of them want to live in mud. So you've got to figure all that out first before you can understand, okay, and are they also responding to really low oxygen because people are over fertilizing their lawns and all that, those nutrients are running into the edge of the lake or into the stream? Are they responding to too much dirt being in the stream and filling in all the rocks so they literally the bugs have no places to live? Or has there been a chemical spill? So it's a sleuthing exercise. And for me, that's what makes it really fun. I've got to think like a bug. I've got to figure out, okay, what do they want? Where do they want to live? And then, is something happening? So how do we ask the bugs if they're doing okay? Well, one of the main things we do is we look at what kinds are there and how many different kinds of critters we find. So this is called richness. So if you find, 10 different kinds of mayflies or 3 different kinds of caddisflies or so forth. So that's the richness. So it's not just how many there are. There might have been 50 of one kind of mayfly. We don't rely so much on absolute numbers as how many different kinds of things we find and the relative abundances, the percentages that things make up in the community. We also can look in the scientific literature and find out if things are sensitive or not so sensitive, pretty tough. So this little mayfly down here, look at those delicate gills on this little mayfly. Not going to do so good if there's a lot of sand and mud in the water. It's going to clog up the gills, make it not easy for this little guy to breathe. Dragonfly, gills are on his side his butt. Really protected, rectal breathing. Your kids will love this. They pump water in and out of their butts in order to breathe, and they can do jet propulsion. If you've ever tried to catch a dragonfly larvae and it squirts away from you, jet propulsion. So remember that. Dragonfly larvae are so cool. So we can say, okay, this guy's tougher. Gills are protected. Big sprawly legs keeps him from being buried in the mud. So he can handle mud, right? So a couple clues right there just by looking at what they look like. We can also look up how they eat. So predators, a lot of times top of the food web guys, the predators, they take it on the chin more than the ones that can chew on leaves or eat whatever. So that's another clue. And then I talked a little bit about how they live. They want to live in rocks. Well, if they want to live in rocks and all the rocks are buried, they may be gone, right? So lots of clues that we can use. So one of the things I really want you to think about with your class is we can learn a lot about physiology, biology, and ecology just by studying the critters. So one thing that comes to mind right away is, how do you breathe? It's really hard to breathe in the water. We can't do it. And feeding, movement, and habitat, I'll be interspersing in the rest of my talk, but I really want to talk about respiration just because it's such an obvious one, and it gives us clues about what's happening, because a lot of things that happen in aquatic systems have to do with reducing the amount of oxygen, and that gives us clues. So some insects are just, and other bugs are just like, screw it, you know what? It's just easier to breathe air. Just gonna breathe air. So mosquitoes, they actually breathe air. They have these things called hydrofuge hairs that repel the water, and they can go right up to the surface and create a tunnel with these hydrofuge hairs from the air down into their respiratory system. This beetle, carrying aqualung, man. He's got hydrofuge hairs underneath his wing covers, called elytra, these wing covers. So he's got an aqualung underneath there. This rat-tailed maggot, don't you love that name? The rat-tailed maggot can live burrowed down in the mud where there is no oxygen because it has this long tail that has, you guessed it, hydrofuge hairs, making a tunnel all the way from the surface down to the rat-tailed maggot himself. And this fly uses these spiracles that are sharp hollow tubes to stick into an aquatic plant and steal air from the plant, because the plant has to get oxygen down to its roots, and so it has oxygen channels down through it to get air to its roots, and this guy steals that oxygen. So if you see, so I should back up. So if you see a lot of these, oxygen's a problem. These guys are all breathing air from the air. They can't even find oxygen. Other ways that things get oxygen. Look at this red. What in our body looks like this? Our blood, hemoglobin. So hemoglobin's super good at hanging on to oxygen when it's at low concentrations. So if you see a lot of really red things, or your kids find a lot of really red things, low oxygen, right? So this is a worm, it's a fly larvae. And then behaviors. So I said they don't move fast, but they can move to better areas. And some of them, like these stoneflies and mayflies, if things get really bad, they'll just let go of the rocks and drift in the current in the stream to get to a better location. Now this stonefly has its gills in its armpits. These tubule things here are its gills. When it gets stressed, it'll start doing push-ups to get more water moving past its gills. Your kids may see this in their leaf packs as they start to sort their leaf packs when the critters are alive, and they may see these guys doing push-ups. It means they're getting stressed, they're running out of oxygen, so you might want to find a way to like put an ice cube in the water, because cooler water holds more oxygen, and that'll give them more oxygen. All right, so what do I do for my job? I get to run around. Well, I used to before I got so old. I get to run around and sample invertebrates. So this is a sampling device. Yes, I'm using a toothbrush to scrub the rocks. Sometimes we use even bigger brushes to scrub all the invertebrates off the rocks. So this is a big fancy sampling device. But we a lot of times just use these simple nets, and there's instructions in the folder that you were given access to on how your kids can make these nets out of a broom handle, a coat hanger, and fiberglass window screen, which is reasonably cheap, and some fishing line to sew it together with. So your kids too can go out in streams and do this kind of thing, or in wetlands. So once we get the bugs, then we look at them, try to figure out what they are, and one of the things we try to do is, as I've said, we try to figure out, are they more sensitive or are they more tolerant to stressors? A lot of the classification of whether or not they're sensitive, kind of sensitive, moderate, or tolerant is based on their response to dissolved oxygen and warm water temperatures. Because remember, warm water temperature means there's going to be less oxygen in the water. And if there's lots of algae in the water, as it dies, the bacteria are going to chew on it. They use oxygen, that takes oxygen out of the water. So you see lots of algae, you see warmer water, you're likely going to have lower oxygen. So all these critters on this page are really tied into needing high oxygen. They like to live in what we call the riffles, where the streams burble through the rocks. That burbling puts a lot of oxygen in the water, and that's where you find these guys. They're living in amongst the rocks. So we've got a couple of stoneflies here. These guys are actually predators. They're crawling through the rocks, chasing after these little mayflies here. And things are not to scale. So the stonefly is actually bigger than the mayfly. And look at the gills here. I showed that before on the mayfly. You know, that's a clue. Needs clean water. Oxygen's really important to this guy. This mayfly, I wish you could see this edge on, but edge on, these guys are so flat, they look like pieces of paper stacked together. You can just barely see them edge on. And that's a clue that they're adapted to living in high current situations and crawling in really small spaces amongst the rocks. So they can get down right to the rocks and the current just flows over them and doesn't wash them away. Or they can crawl in the cracks to try to avoid those stoneflies, right? And then this in the center is a type of caddisfly. And so I say that these are sensitive invertebrates. We think of just all the stoneflies, all the mayflies, all the caddisflies as the more sensitive invertebrates. Unfortunately, that's not entirely true. There are always those special species in each group that can hang out in tougher conditions. But a lot of these caddisflies, and many of them make special cases of sand or sticks or whatever sewn together with their silk. A lot of times you can identify what they are just by the shape of their case. So this is one that's more sensitive, has little gills here. I think you can see the tubules on its body. But some are more tolerant. And so now let's talk about the more tolerant individuals. So again, hemoglobin's really a clue. So if you see this bright red, low dissolved oxygen. Now, remember to tell your kids, if you put them in isopropyl alcohol to preserve them, it denatures the hemoglobin and the red color will go away. You can only see this red color while they're alive. So you want to make note of that. Here's a mayfly that's more tolerant. Look at this. It has gill covers that protect its gills. So these are like pads that just protect the delicate gills underneath, and in fact you can't even see them, so it can tolerate more sand. And in fact I found this one in I collect these all the time in sandy streams. I talked about the dragonfly and the gills up its butt to protect them. And think about the water striders. They're like, you know what, we're just going to breathe air, we're going to be up on top, we're going to stay out of the whole thing, don't really care what's going on underneath the surface. So again, you've got all these different kinds of clues about these invertebrates that if you find a whole bunch of them and not much else, maybe oxygen's a problem in that area. Now, if your students put their packs in ponds and wetlands, you're going to get more of these things that are tolerant of low dissolved oxygen, just because ponds and wetlands often have oxygen stress overnight. Because they're more productive areas. That's a natural thing. It doesn't mean the habitat's bad. It just means that's a harsher condition to live in naturally. So as you talk to your students about where they want to put out their leaf packs, get them thinking about the types of habitats that have water in them and where they might want to set their leaf packs out. So even in a stream, there are different habitats. I've drawn this green line here, divides the stream into two parts. Over here is the slower water. You see all the vegetation growing up. There's more mud. Things can burrow in the mud and climb on the vegetation. On this side, we have one of those riffles where the water's burbling through the rocks. The current is faster. You have the things that we call clingers. They cling tightly to the rocks so they don't get swept away. A water penny beetle is one of the ultimate clingers. The caddisflies, some of them can even attach themselves. They spin the silk and spin themselves right to the rocks so they can't be washed away. So two different habitats right here in the stream, right next to each other. Edge of a pond is going to be a really different habitat. Edge of a lake is going to be a different habitat. An inland wetland is going to be a different habitat. And you're going to get different kinds of critters in all those places adapted to different living situations. So it's a great chance to talk about what are the adaptations that allow the different critters to live in these various places and do comparisons across your class as they take pictures of what they found. So I'm going to use an example to try to illustrate how you can think about doing this with your class. My example is all about streams, but you could do this with, you know, stream versus pond versus wetland or whatever your class has available to them. So on the left here is a creek up north by Duluth, up north by Tofte, up the north shore of Lake Superior. That is super bouldery. I couldn't find dirt. I dug down in this stream trying to find the stream bottom where I get to like dirt and mud. Never found it. I had my head underwater. It's crazy. There's so much habitat space here because there's all these rocks and the critters can just live all among the rocks. And then there's this poor creek and it's not doing so well, right? Muddy and really slow moving water. The mud in the water allows the water to warm up, so it has lower dissolved oxygen. This is just outside of Two Harbors, just north of Duluth. So what's not important is where the streams are. What's important is the different types of habitats. Really rocky, nice system, lots of flow, lots of habitat space, lots of oxygen, and this poor creek. So in a rocky system, and I had to use pictures from other streams because I just didn't have this all in one package. So again, ignore the fact that they're from different streams. What's important is this is from a rocky system like the one I showed you. And what we see are lots of really sensitive stoneflies, mayflies, some caddisflies over here, and here's another turtle case caddisfly. But we have a few of the less sensitive things. Snails, leeches, I think these are some big fly larvae, some wormy things. For right now, we're just gonna call them wormy things. So in any habitat, even if it's really good, you're gonna have some of the more tolerant critters. So it's the proportions that matter. We have lots of things that need high dissolved oxygen in this picture, and not a lot of things that are super tolerant. Now, here is a system that's kind of an in-between system. This is a sandy stream. It's over on the Bayfield Peninsula of Wisconsin along Lake Superior's north shore or south shore. And it's in an area where there's just lots of sand. The sand buries all the rocks. There's just no rocky habitat space, but the water's cold. It comes out of a sand aquifer. Water's like 50 degrees all year round. Lots of oxygen, not a lot of habitat space. So the critters instead may live attached to all these leaves and twigs and things that occur in the streams, or they're just really good swimmers. So what do we see? We have some mayflies here. These guys, they swim so well, they're called little minnow mayflies. If you saw them swimming in the stream, they swim so well you would think you were seeing tiny little fish. But you don't really see stoneflies here. Not much for caddisflies. Lots of wormy things here, wormy things over here. These are fly larvae. This fly larvae here is a crane fly larvae. Your students will likely find some in their leaf packs. More clams. These are aquatic sow bugs. You've seen them on land. You call them pill bugs, and in terrestrial, when the kind that live in terrestrial situations. So it's kind of an in-between stream. And then our poor muddy systems. These clay streams occur along the south shore and in parts of the north shore of Lake Superior. Again, all these things were taken within 100 miles of Duluth. These muddy systems, they just don't have enough stream power to wash out the mud, and they're flowing through old lake clay plains, so there's just tons of clay. And look at what we find. Lots of clams, lots of wormy things, some beetle larvae, couple little mayflies. These are the more hardy caddisflies that I talked about. These caddisflies, they can pretty much take it. They spin nets that would have tiny little nets. They catch tiny little particles of food on them. And the caddisflies just go out and eat the food off their nets. They live inside the back of the net to protect them from fish or whatever would come along to eat them. But, you know, lots of wormy stuff, lots of tolerant stuff. So you can tell this site, just on the grand scheme of things, is a harsh site. Now, did humans cause this? I mean, maybe a little bit with logging and some of the stuff we've done, but by and large, these systems are naturally harsh. So another thing to think about with your students, just because all their invertebrates come out in the tolerant category, doesn't mean that humans screwed up anything. It just means that there are naturally harsh places out there in the world where these critters have to live. So now, how do we use the data? So I hope these aren't going to be too intimidating. I'm going to walk you through these graphs. So one way that I like to really visualize the data is to try to stuff all of it into one column, which is kind of scary on a bar graph. But what I've done is I've taken all the rocky streams and I've averaged out their invertebrates. So this is an average of all the rocky bed streams. And all I'm plotting is how many of them there are, how many critters I found by the categories. So you can see all the stoneflies are in blue, dragonflies and damselflies are in these yellowish colors. And then we've got leeches down here in green. And first of all, you'll notice that those clay and sand streams just don't have as many critters. It's just a harsher environment. They just can't live there. But, you know, relatively higher proportion, much higher proportion of leeches and much lower amounts of the stoneflies and the more sensitive individuals. So you can, you know, do this kind of comparison with your students' data. And you can make, like, if they're in a rocky stream versus some other kind of stream versus a pond versus a lake, and just show how they're different. Another thing you can do is once they've figured out what their critters are, and you can put them in the categories, and there's a data sheet that Marty's going to show you that helps with that, of sensitive versus in the middle versus the tolerant types of critters. And then again, you can say, well, how many different kinds of things, so number of taxa, that richness, how many different kinds of critters were found in each of these different types of habitats? And again, it clearly shows the sensitive taxa do a lot better in the rocky streams than in the clay streams, with the sandy streams in the middle. Conversely, the tolerant, there's a lot more different types of tolerant individuals, and this is by percent of the community now. 30% of the community, and these are error bars, so up to nearly 50% in some streams, of the community was made up of things that we would consider pretty darn tolerant, versus in a rocky stream, notice it's not zero. There are still tolerant individuals in those rocky and sandy streams. They just make up a much smaller percentage of the community. So, and for those older students, calculating percents, right? So throw a little math in here. This I know looks really intimidating. So let me walk you through it, because you guys can do this with your students. Each bar is all the critters found at one site. So for you guys, this might be, each bar might be each student's data, all the critters they found in their leaf pack. So all we've done is do it by proportions. So in this first example, there's what, 30% mayflies, and then maybe 15% stoneflies and 10% caddisflies and so forth. You create a stacked bar of all the different things you found, and don't worry, you can just lump stuff into other. So these are all the sites I've sampled around Duluth, the North and South Shore area. And I'm highlighting that rocky stream I showed you at first, that really rocky one where I couldn't find the bottom, and the muddy stream that was horrible. And I've ordered from left to right these streams by the amount, the proportion of our most sensitive critters, the mayflies, stoneflies, and caddisflies. So highest proportion of the community down to lowest proportion of the community. And you see that poor muddy stream, it's way down there. Whereas that rocky stream, not the best one I've ever found, not the one with the most. that's this stream over here, but still, you know, more than 50% of the community in those 3 most sensitive categories, right? So a great comparison, you could do this across, you could lump by category. So you could say, okay, we'll take all the ponds and everybody who's in the pond sample will compare them to each other, because it's not really fair to compare ponds to streams. Ponds are never, ever going to look as good as a stream. Or wetlands, same situation. A little bit simpler way to do this would be just to lump all the sensitive taxa together and just count how many different types of sensitive critters you have versus everything else. And so again, this is, if you can think of each bar as being 1 leaf pack sample, and I've again sorted it so that it goes from the greatest number of different types of sensitive critters to the least. And I'm highlighting our rocky stream and our clay stream. So again, that shows, you know, that rocky stream wasn't the best, didn't have the most, but it's on the top end. Whereas that poor clay stream down here at the end, what's amazing is stuff lives in it all, right? Life finds a way, as one of the movies said. Okay, so we're gonna stop there for today. When I come back and talk to you again in 5 or 6 weeks, we're gonna walk you through how to identify your critters, but Marty's gonna tell you some stuff that you can use in the meantime. So I will figure out how to stop sharing and turn this back over to Marty.
Thanks, Val. We would like to take a couple of minutes because I think that some questions rolled in. So I'm actually going to turn this over to Lori for fielding maybe 1 or 2 questions, and then we'll roll into the activity.
Yeah, all right. Dr. Brady, thank you so much. Those slides were great, and I think they give us a good idea of kind of the work you and your team have been doing in the streams in and around Duluth. Brian wants to know, have you identified any keystone species in your research in streams around you?
Keystone species, so keystone can mean 2 different things.
Sure.
So one is a great indicator species. Another one is a species that sort of dominates and controls the community for good or bad. So in general, in these streams, you know, some of the fish can play that role. The invertebrates tend not to, although some of the invasive crayfish will play that role in a negative way. And so what we don't like to find is like big invasions of rusty crayfish that can really change the shape of a community. But fish tend to change the community in major ways, and so we have to be careful to never compare sites with lots of fish to sites with no fish, because certain invertebrates are really easy prey for fish, and so will never occur with fish around.
Oh, that's a really good point. I hadn't thought about that. So what can you tell me a little bit? I'm curious about your current research. Has your research changed as I imagine it has a little bit, aside from just going from stream to stream. Have you changed the trajectory of your research at all? What are you currently focusing on?
Well, actually, I'm mostly working in wetlands right now. We don't have any current stream research. I do keep collecting stream data because it helps us to figure out, especially with restoration sites. I have a proposal in right now to the state to hopefully be funded to use the data that we have to assess stream restorations and whether or not they've achieved their goals of actually fixing bad stream sites, places with bank erosion and things like that. In wetlands, we do a lot of the same work. I gave the example of streams. We do the same thing with bugs in wetlands and trying to assess how the Great Lakes coastal wetlands are holding their own and if restoration activities are actually making things better in these coastal wetlands. We spend a ton of money on restoration activities, and so we want to make sure that it's actually working.
Yeah. Absolutely. All right. I'm going to ask this one question to Marty. I noticed you just put it in answer, but I'm going to get Dr. Brady's opinion on it. So Zach wants to know, when I do this with a class, would I have better success in the spring or in the fall?
Normally, you have better success in the fall. You can do early spring before the invertebrates or for the insects emerge, but it can be tough because you need to do it right when ice off happens. So if you're farther south, you can most likely, you could try it in the spring. Fall is a more sure time for getting students safely into streams. And remember, safety's got to be key. The caveat is, if you get a bad drought and some of your smaller streams stop flowing, fall is going to be a really crappy time. And you're going to go, oh, I should have done it in the spring, but that's always 20/20 hindsight.
Sure, sure. Thank you so much. That's all the questions for now.
Okay, well, thanks for that really, really great and exciting kickoff to our virtual workshop series. I'm going to now quickly do a little bit of activity demonstration for you folks. We're still intending to break at 9:55, so I'm just going to start rolling through this and see how far I get along. So first, I need to share my screen. We've got that. And then I need to minimize myself. There we go. Okay, so, oh, I'm not sharing my screen yet. Never mind. Those are notes for myself. Okay, so right now I'm going to be talking a little bit about the leaf pack, and we'll get going on that. So first of all, the folks that are in this workshop should have received the onion-like sack that comes with the leaf pack kit. This is just one portion of many items that are in the leaf pack kit, but it is one of the most important ones. And of course, in addition to getting a sack like this in a kit, you can also order them online through Amazon, or you can order them piecemeal through the Leaf Pack Network website. Additionally, they are the same sorts of sacks that you keep onions or citrus fruits in, so they're fairly readily available. The stack then just simply gets stuffed with leaves, although maybe I shouldn't say simply, because there are some leaves that are better than others for sampling. Maple and willow leaves are particularly good because they're softer and they're easier to break down. They're nutritious. Some of the tougher, harder leaves or grasses aren't so good, so something like oak leaves or your grasses or maybe a sedge or conifers are not as good because they're not as nutritious and they're a little bit more structured and tougher to break down. One of the differences that you'll see is that in the Leaf Pack Network manual, the focus, although there are classroom pieces, there's also a real focus on citizen science, and so there are some pieces that they're looking to standardize. And so one of the things that is recommended in the Leaf Pack manual is that you collect dried leaves and use them to make your leaf pack. And Val assured me that that's not at all necessary. You can use green leaves. In fact, she actually encouraged it because they do have a little bit more nutrition in them than your dried brown leaves that are already on their way. So just looking for green leaves will also suffice and make things easier probably for this time of year. So once you've got your sack and you've stuffed it full of leaves, you need to identify a water body that you want to put it in. Val did mention that there are some, that macroinvertebrates basically live in any kind of water system. And so you can identify any water system and put it in there. Within the Leaf Pack network, they do talk about targeting riffled streams that are fairly shallow, like below knee depth. And again, they're kind of looking to standardize where things are collected so that information can be put into a larger network and a larger body of data. So if you're interested in that, do remember that those sorts of pieces are sort of standardized. However, there can be other interesting ways to look at macroinvertebrates and look at placing your leaf packs that sort of expand or relax some of those restrictions, which I think is important now, given that students will probably be in different places and may not be able to be as picky about where they're putting their leaf pack. Once you've identified your stream, your river, your lake, a stormwater pond, a roadside ditch, a backyard pond, you'll want to stake your leaf pack into that area and secure it really well. As you can imagine, if you are focusing on an area where water is a little bit faster moving, like this riffle, you're going to want to be really diligent in securing that leaf pack. So you'll probably want to tie a rope or a string around it, and then maybe anchor it with a rock, and then run that string up to a bank and tie it off to a tree or a stake or something like that, because it's really sad if you've put your leaf pack out and then it gets claimed by the water. So you stake it in there, and then after that, oh, and the other thing you can do is you should have received a label. That label is on a special kind of paper. It's called Right in the Rain paper. And so it is waterproof, and you can submerge that along with your leaf pack. The types of utensils that work best for writing on it are either a pencil or a Sharpie marker. And it's a good idea to label and identify your leaf pack, because then curious people will understand what that leaf pack is for, and hopefully will leave it undisturbed. If they take it, of course, you are out one leaf pack. And so that can be one way of trying to manage it. You could even put, a phone number on or something like that if you're willing to share that information, so people can ask you questions, or if they're curious about why there is this pack of leaves that is put into a body of water. So then you just leave it, for 3 to 4 weeks. And so you still have a couple of weeks to identify a water body, and then you figure out the best place to stake it. And then after that, you might want to check on it, once or twice to just make sure it's still in place and that kind of thing. But when you check on it, one of the key tips is just look at it. You don't want to, you need to resist the temptation to sort of move it or poke it or shake it, because you can imagine the things that have decided to crawl in there may leave if they detect a threat, which is the thing that they may consider you if you're poking at the place they've decided to reside. So make sure to just monitor it like that. When it's time to take it out, be quick. Have a bucket or a dishpan or something right at the ready that you can very quickly transfer your leaf pack into. So when the macroinvertebrates sense that disruption and they try to dart out, the only place they're darting is into the dishpan instead of back into the stream. The other really key tip here is don't let it dry out, since of course they're aquatic. If you let the leaf pack dry out, they will dry and die, and they'll be a lot harder to identify because they will desiccate or dry out. There are notes about this information and more that are in the Google Drive. So if you haven't been able to keep up with the information that I've shared right now, don't worry about it. That information is already populated for you also in your Google Drive. So once you have actually created, I've really got to get cracking here. Okay, so once you have collected your leaf pack, there are a couple of observations that can be made really quickly. What does the leaf pack look like prior to being added into the stream? Does it look the same? Does it smell the same? Are there things that seem to have changed on the leaf pack since it was first put in 3 weeks ago? More than likely the answer is yes. There's probably some slimy stuff that's on the outside of the leaves now. And this, of course, is bacteria. Also, algae and fungus or fungi can populate the outside of the leaf. And this is a really natural thing that just is part of the nutrient cycle. That bacteria is breaking down all of the nutrients that are in the leaves and making it available to other organisms. Additionally, there's other sorting that can be done. Animals can be sorted from non-animals to sort of get your youth and your learners thinking about what it means to be an animal versus what it is to be not an animal. You can also look at organic materials versus maybe some non-organic materials that may have gotten collected by being swept into a stream if you're in a place that's got some flow to it. And then you can look at macroinvertebrates that are young, macroinvertebrates that are more mature, if those two are present, and have conversations about things like incomplete versus complete metamorphosis. Of course, the ultimate level of sorting really is about taxonomy and being able to identify some of the organisms so they can tell you something about the water body that you have pulled them from. And there are a couple of tools that I would love to show you. I think that they're fairly easy to navigate on your own, and so in the interest of time, I'm not going to show you them now, but again, this resource is found in your Google Drive. It's called the macroinvertebrates.org website, and it's a really wonderful tool for helping with identification. Additionally, one of the things that comes with your leaf pack kit is this right in the rain paper sorting piece right here. And I recognize that not everybody is going to be getting one of these kits. They run, I think, about $260. There is one that's going, at least one that's going to be available for a drawing. However, what I did, recognizing that we wouldn't be able to have everybody have access to that, is I went ahead and created the starting of a PowerPoint template that you guys could use. And so right here, you can see that I just created this. It's still all able to be manipulated. So you can go in and add line art if you want. It's color coded to indicate sensitive, somewhat sensitive, and tolerant organisms. And so then there's just a spot for you to place them. These will print out on 8.5 by 11 sheets of paper, and those circles are approximately the size of a Petri dish. Of course, if you don't have a Petri dish, you can use, you know, the jar, a lid from a jar or something like that to hold all of your precious macroinvertebrates that you've been looking at. So once you've collected them, sorted them, and done some identifying, you can actually work on something called a biotic index sheet. And this biotic index sheet, I'm not going to go through in a lot of great detail because we are running kind of close on time here, but I will just kind of walk you through the steps. So you can organize your organisms based on sensitivity levels, and so that's your first step, and then you simply count up the organisms and you put them in the column. And then you do that for each of the categories, somewhat sensitive and tolerant. And then you simply sum the number of individuals, and that's right here. But the more important part actually is counting up the number of taxa, which equates in each group, which equates to simply counting up the number of taxa that are present in each of these. So you just each row, stoneflies are a taxa, mayflies are a taxa, caddisflies are a taxa. And so in this case, we have 7 of the 8 rows here that have a number associated with it. This one, Dobson flies, does not. You do the same for somewhat sensitive organisms and for tolerant organisms. Once you have that number, you add across this way. And so you have 21 plus 10 plus 4 equals 35. And this is sort of where the magic happens. And you're able to actually take a look at what macroinvertebrates can tell you about the water that you are working in. Maybe it's high oxygen, maybe it's low oxygen, that kind of thing. Right here, you get your pollution tolerance index rating. And in this case, it is excellent. Now, one really important point of clarification with this is this biotic index data sheet is set up to go with the citizen science portion, which has some of those constraints on it. That constraint being that this is really set up for what does it look like in a stream that has riffles. And so if you go into a different kind of water system like a lake or a pond, you may end up with something that looks like a lower pollution tolerance index rating, which may tell you, or think it may tell you, that that is not healthy water. And as Valerie pointed out in her presentation, it's just a different kind of system, and it doesn't necessarily mean that it is polluted. And so with that, I am going to stop my sharing and get back to our regularly scheduled programming. So that, I think, is it for me, and we need to roll right into our break. And so, Anne, I'm going to ask you for a little guidance here. How long shall we allow for that?
Let's reconvene at 10:03. That gives people 5 minutes to visit the bathroom or grab a cup of coffee. So back here at 10:03, please.
Welcome back, everybody. So we are going to be setting up now to go into our small breakout rooms. And so we have we've got some folks that are getting broken into that room, and then we do have some people who are going to be taking some notes. I think everyone has the link to a Google form, but there's really only one person per group that needs to be filling that out. And so that's what I have for stage setting, and I encourage any of the other planners to hop in if there's a piece that I missed. And if not, then we can just get popped into our breakout rooms?
Each room will have a facilitator, and I'm going to send you to your room. You'll be arranged basically by like age group. So hopefully that will give you opportunity to network and to work together. So please accept when I open the room, you'll be asked to go to the room, and please say yes. All right, have a good discussion.
Okay, so after our quick breakout session that we had, we did have a couple of good questions in our group, but we wanted to take a little bit of time to just wrap up what we've learned today. And so this was not a thing, actually, that we even addressed in our breakout room. We did not get that far, but I am curious to open it up to all of the breakout room folks to share maybe one thing that you guys discussed or that you learned during your breakout session. And so if we could do that, that would be awesome. And so we've got, how many breakout rooms do we have, Anne?
The main room became a breakout room.
Okay, and so why don't we just start with those folks that were in breakout room 1. What is a thing that you learned or discussed in your breakout session, and can you please share it with the group? Maybe the person who took notes can be the one who shares.
So we had a discussion about the different ways that we could use them. We came up with some writing projects from the perspective of the bug after the students had studied them to talk about their home. We talked about that one of the groups does fishing expeditions already and potentially tying it to that. We talked about potentially doing some sorting and graphing and bringing out a dissecting microscope that no one has used for 5 years, anticipating that there won't be field trips, but the students potentially could place the packs on their own, making packs of similar form from what the teachers have been giving, and asking people now to start saving their onion and orange sacks, so that might be a possibility in the future.
Awesome. That's a fantastic summary. It sounds like you guys had like the most productive breakout session. So group number 2, who took notes, and can you share out?
Yeah, hi, that was me. So I thought the most interesting part of our discussion was talking about how we would do this online or in hybrids. And some of the teachers were talking about creating leaf packs for their kids and putting them in the packs of things that kids would pick up if we are not back in person this fall. And then have the kids do the collection, and then have a Zoom, like make it a family affair. So have the families take the kids out to a body of water nearby, place the packs, go back, collect them, bring them back, and then do a Zoom discussion about what they found. I thought that was a pretty good idea. Another person talked about adding this to already existing discussions about like water quality in streams. And yeah, so that was the main thing.
Okay, thank you. And then number 3, that was us. So we had just a brief conversation, really more about taxonomy and the different kinds of guides that are out there. And so we had some folks in our crew that have done quite a bit of work with macroinvertebrates already, and then we also had some who are just kind of getting back into it, and so we're really welcome and open to learning about some of the resources for getting going. And so I shared the macroinvertebrates.org website as an online tool, and I'd mentioned that before. And then Dave Johnson also showed one of the sheets that he uses when he does his work in Duluth, and that is I can't think of what the acronym stands for, but it's the key that the University of Wisconsin Extension uses for their work. It's pretty well known in these areas, and so all of those pieces were sort of talked about. So we really focused on the taxonomy and a little bit on methods too. Dave had some questions for Brian about how he did zooplankton collection with his students. And that's my my summary for group 3. So group 4.
So that's me. And our group, we covered a lot of ground in a very short time. And I think the things I'd like to mention are It was like a no-brainer when we said, could you use this, you know, in an online or hybrid format? The same thing that we already heard, but also a strong emphasis on the data that come out of this would be awesome for helping the kids do better with testing, giving them a chance to graph, do simple calculations. And I think that's a huge part of this, is how do you quantify working from home, right? Get all that other stuff in there. The one other thing, I wanted to mention was we had questions about, is there some way that one of our teachers wants to combine this with storm drain program she's working with? And is there some way to sample for invertebrates in a storm water system? And is that a reasonable thing to do? And so Val, that might be something we could, you know, chat about or somehow get back into. There was a ton of other stuff, but I wanted the other groups to have a chance. So we'll just stop with that.
Okay, thank you.
One thing, safety guidelines. I did want to say safety guidelines. Somebody comes up with a good idea for a list of safety guidelines for the kids and the parents. If it's hybrid, share them with this whole group. You know, if you're creating something, share it. Done.
Thank you for that awesome and succinct summary. So group number 5.
Sure. One thing we did talk about, just kind of play off that, we did talk about safety. And if you didn't see in the chat, Val did kind of share some of the recommendations that she shared with us in our breakout group. But for those that maybe missed that in the chat, definitely recommended to have a buddy. So like if you're going out within classrooms, obviously you're probably putting kids out in groups that will have buddies. But if you're doing this distance learning, make sure you're insisting that they have like a family member buddy, you know, kind of depending on what the distancing guidelines are at that time, but you know, it should be not, you know, sending kids out on their own with that. Also, she let us know that you could get the bag into the body of water without actually going to the body of water. So if you put a rock in the bag and then tied like a rope onto it, you kind of toss the bag. You know, as long as you're holding onto the one end of the rope, you know, send the bag tied to the other rope into the body of water, and then later you'd be able to reel it out when needed. So if they couldn't get into a body of water safely, you know, obviously that's not an ideal situation because in the time it takes you to drag it back out, some of the critters are getting out, but you know, it's better than nothing certainly, and you know, keeping kids safe is good. As far as how we would use this, you know, with the Minnesota state standards, you know, with a big emphasis on just the practices of science. I mean, there's tons of opportunities for kids to design their own experiments and then to analyze the data. So those are huge things there. And then the kind of last thing we touched on was just, you know, looking for good student-friendly places for info on macroinvertebrates. So, you know, just that aren't, you know, as, you know, in student-friendly language as possible. It's just things that we want to make sure if people have those get shared with everybody and just have interesting story like butt breathing and everything else. So that was group 5.
Great. Thank you so much, Eric, for that quick summary. And group 6.
Yeah, okay, I was group 6. We talked about with adapting to different grade levels, we had discussed adding more factors like statistics and more complex math and that sort of a thing with older grades. And then we also discussed adapting different methods of teaching vocabulary for the online environment. And we talked about pre-teaching vocabulary. And then we also discussed the value of potentially taking pictures of what students find in their leaf pack, so then they can compare that with their classmates and then compare between the different ecosystems that they have around their homes. So yeah.
Oh, it sounds like we had so thank you for that, Brenna. It sounds like there was some really great conversation that happened in all of the rooms, and I hope this mini share out gave all of you at least, you know, one other good idea about how to maybe implement this with your students. And so
Marty, can I interrupt? The main room did have some conversation, so could we invite the main room to chat too?
Oh, of course, please.
Thank you.
Hi, we were the ad hoc, our invite didn't come through group. Our conversation was just about how this is, and same as a lot of other groups, how this is perfect virtually. that if you had to do homeschool, you had to do some sort of virtual, that this is really a perfect example of citizen science and hands-on. I was really intrigued. John from St. Paul said that they actually raised trout in their classroom and then released them. I just think he needs to share something about that at some point. I'm putting him on the spot right now. That sounds pretty cool. And then Olivia said something about saving, not feeling like this has to be all done within a couple days. So saving it, that you can do the data collection at one point and the picture ID at another point, and then maybe some conversations about food webs at another point, and just sort of, it's okay to spread out the process because the kids, it is overwhelming to try to do all of the IDing and data collection and graphing and everything so quickly. That's probably it.
Awesome. Well, thank you for that share out too. And thanks, Anne, for reminding me that there was a group that didn't have a number. So, and I did see one reaction. I saw one thumbs up as Allison was talking. So in case you didn't all see that, there it was. So I think at this point, this concludes our workshop. And I would like to invite Anne back for any instruction on how we're transitioning between Joyful Hour and the workshop that we had. So I know some of you will be leaving and others will be joining. So just thanks so much for being a part of this. If you are dropping off now, we really appreciate you being a part of the first portion of our virtual workshop series. And we really hope to see you back again at other workshops. So that's what I have. I'll turn it over to Anne.
Great, thanks Marty. Not much, we're gonna have another 5-minute break where hopefully I can set the rooms up this time and make them work. So just remember when I do set up the rooms to accept to go there. We'll have a 5-minute break and then a quick introduction to what will happen in the room, and then you'll continue on. So I think if you didn't sign up for the Joyful Hour, you can stay. It might just take me a minute to fling you in a room. So take a break, and I do want to say a special thank you to Val and her student Holly for coming today. It was great to really hear from them. So thanks a lot, and we will reconvene, let us say at 10, I can't do my math, 10:37. All right, so we're going to have the same idea of going into the breakout rooms, and in each room, hopefully there'll be someone who can talk with us about the Denis Sullivan shipboard experience. And there should be a facilitator in each of the rooms. And again, if you look in the chat, I'm hoping that, yep, if you look in the chat, Lori, our chat monitor, has added a bit.ly that leads to dragonflies. And so that will be the form where you can take notes. Let me check the schedule and see how long we're supposed to be in the room. It looks like we have about 30 minutes or so, maybe even close to 40 minutes. So really, this is a chance for you guys to, we've heard over the years that many of the teachers really like to have time to network and to brainstorm. So this, please think about ways that not only you might consider something like a shipboard science workshop like the Denis Sullivan, but also ways maybe perhaps people in your room, you can work across borders, across school districts. One of the goals of the Center for Great Lakes Literacy is to build this community of practice. So maybe bring out your action plan could be also another opportunity to share, kind of continue. We, in the main room, we didn't have a long time to talk about the leaf pack. So that would be another thing you could begin to talk about. But I don't doubt that there won't be great conversations. So I will be giving a 5-minute warning again, and also hopefully I'll give you 60 seconds to close the room. So you see the dragonflies bit.ly link, and so I'm going to send you guys to the room, and my fingers are deeply crossed that you all go somewhere. So we'll see you in a little while, and every room has a facilitator, I think. Yep, so they should be able to start the discussion. All right, hope you have a really great breakout. Bye.
But anyway, everyone, welcome back. I hope you had a good workshop and breakout discussion. I think we are on for another 12 minutes, and so I think I'm still not. Maybe one of the other planners can help me with orientation here.
I have an idea.
What's your idea?
How many people who have not been on the Denis Sullivan want to go sometime? Raise your hand if you can find the little hand thumbs up thing.
Haven't been?
So you didn't get scared off if you have not already been. I was just curious if anybody got scared off, you know.
That's a good way to start.
I'm looking.
Marty, do we want to have a share? Do we want to have a share out for each of the groups?
Yes, we do. I know that was one of the things we were going to do, but I was not sure if there were other things. So let's start with the share out. Yeah, I was gone somewhere, but anyway. So welcome back. I'm really excited to see that there were so many thumbs up based on the question that Cindy posed. Yeah, the Denis Sullivan is a really wonderful vessel, and we can share out what we talked about in our group, but why don't we go ahead and start with the, well, let's go backwards this time. Let's see, we had 5 breakout groups, or Ann, did some folks stay with you again?
No, we had 4 breakouts.
We had 4 breakout groups. Okay, so so let's start with the 4th breakout group and just learn, what did you guys talk about? What'd you learn?
I'll just start, but I want other people to echo or jump in because I think we started with the Denis Sullivan is incredible. You will take a lot back with you, but it's not your classic teacher science learning experience. It's there, but it's a lot more than that and a lot different than that. And then I would maybe turn to Brian, Mick, if Olivia can talk now, just to maybe add a sentence to that, and then hear if our, well, I know that our educators, at least 2 out of the 3 are really excited about the concept of going on the ship sometime. So how about Mick, do you have anything you want to add to that? We went in a lot of directions, so.
Yeah, it's a great experience. I've been on a bunch of different ship stuff, and like Cindy said, it's way different than all those. It's the science education piece, but then it's actually like being part of the crew, which it takes it to a whole different level.
Mm-hmm. Brian, anything?
Yeah, I guess from a teacher's perspective, it really gives you a whole rounded experience, not just with research itself, which there isn't a lot of focus on, but there is some. But it's community, it's collaboration, learning, working with people, and really working as a team to sail this huge ship, and which I knew nothing about. I knew nothing about sailing, and as overwhelming as it was, it really became a good experience for me, and I'm glad I would do it again in a heartbeat. So
that does make me want to share one thing that Brian brought up that I thought was really telling. Was he felt like, he said, I have a better empathy with my kids that I teach now, because they come into a brand new subject and they're nervous, and I'm paraphrasing, he didn't say it quite like this, but I really got a feeling for what it feels like to be a kid in a new class. I thought that was really important. Olivia, I don't know if you can speak, or even if you're still, but Olivia shared a lot of good stuff that's in that chat, so hopefully we can share it. And lastly, before we turn it to the next group, do any of the 3 who are in our group have something they want to share about what they heard and how it affected them? So that'd be Nicole, Leah, or who is our 3rd person, Molly. And just quickly, we don't have a ton of time, turn it over to the next group. Nicole.
I guess I just liked that they had a whole experience. It wasn't just the science, or it wasn't just the fun, but the memories. I liked asking that, because when we love a place, or when we feel like we love a place, we'll take care of it. So I like hearing that.
Yep. Okay, and I don't see either of the other 2 unclicking. So I think that would be our group report unless somebody has something they really want to add. Thanks.
Okay, thank you. I would say that that's all really good and so accurate information, because we a couple of us have been fortunate enough to be on the ship several times, and there's been some change in the programming over the course of it. In our group, we talked a little bit about just what it's like to be on that 5-day sail and to spend time working on the ship and learning some of the water quality and the science, but then also expanding that into some of the other topics that we've covered and some of the other it's not just for science teachers. Last year we had some teachers who were on that were had a background in political science, and we have people who are more focused on English, and all of those pieces I think can be important for expanding Great Lakes literacy and working on working in these programs. And so we talked a little bit about that. And we talked a little bit about, once we're done with our, workshop, how do we bring it back? What do we do? One person mentioned the limnolone program and using the Hydro Lab. But I would also like to take the time to offer up for the people that were in my group to share a little bit as well. So I will turn it over to the rest of my group mates and see what they might have to say.
Now, something we didn't talk about in the group, but I want to echo what Brian said, is that appreciation for what it's like to do something completely new and completely out of your comfort level. I mentioned it was kind of a life-changing event. I think that's what really got me, was just doing something that I've never even thought I'd have the chance to do before. So I think that was almost more valuable than the science itself, just having that experience.
We also had Lisa in our group, and Lisa was able to speak about the Denis Sullivan from a different perspective because she has worked at Discovery World. And so, Lisa, did you have a little to add?
Just that it's such an important way for kids to get a sense of their environment and just a better sense of space. So I think if you can bring students on to some of their programming at Discovery World, it's really powerful. And they do half-day programming or full-day sales, so it's more accessible for students. And then anything that we would learn as educators, I think you can modify some of those experiences for your students too. And I think it's powerful for them to see you in a new scenario, in a different comfort zone than you're used to, and see how you overcome that. And that gives them some confidence in how they can accomplish those things too.
Thank you. And MJ or Joy?
I just wrote down a lot of ideas how I incorporate what I've learned with art and careers, and also excited to hear about the sailing program, about thinking to apply.
Cool. Well, I think that that's it for group 3. On to group 2. It's occurring to me that we might not know what group 2 is.
I'm here.
Oh, okay.
Somebody muted me. You can't shut me up. Okay, so, well, we only touched a tiny bit of the whole experience. It's not enough time, but we did have some really interesting ideas. We passed around a lot of really great ideas. Some of the people in our group have had the kids on the ship for a day, and that was really interesting to me, because maybe that's something we could pull off with the right grant money and enough time. Maybe not this year, but working on it. We talked about how even for kids that live in the cities that are on the lakes, so few kids have not actually experienced the lake. Not, more than just seeing it from the highway. I mean, actually, exploring it, being on a real, lake shore. not necessarily just the beach, but some understanding of what these amazing lakes are. I mean, we're in St. Paul, so we're like 2.5 hours away. We could get there. We have in the past. Some of our kids have come from cities like that, but I think Liz, I think you said it was like 60% of the kids.
Yeah, we find that of even seeing, so I'm from the School of Freshwater Sciences in Milwaukee, and I take kids out quite a bit, and we pull them regularly. And when I take them to the shore of Lake Michigan, I ask them, how many of you have been here before? And only about 60% of the kids have.
Yeah, that's amazing.
And that's, even with the kids who are living in Milwaukee, it's there are some interesting barriers and equity type things that happen in the area.
Mm-hmm. So that was really interesting. What did anybody else have to say in our group? I don't want to say everything. Oh, maybe they're not here. Oh, there she is.
I think we emphasize the fact that we have to be very broad in our thinking of where these water sources would be. That it could be a natural stream, or it could be a city, creek bed, or it could be, like you said, the ponds that you use, or the stormwater collection that, forget who else mentioned that, but I think that's important.
And also that those local things can easily be tied to the bigger systems of the Great Lakes. You might not even be in the watershed, but doing the local work with your kids gives them a hands-on appreciation, and then it's easy to tie that to the bigger picture and even global.
Having said that, I'm thinking of the fact that, especially for doing distance learning, and we will be emphasizing use of technology, are there any members here who would be interested in maybe creating some sort of document where we pinpoint places we got water to share with the larger group, so that, like my kids in Milwaukee would know that that we're all collecting waters from different spots, and that eventually these waters are all going to and coming from a shared place?
I think that's a good idea. Can we facilitate like maybe a sign up of who wants to share, just even a Google Doc or something like that, hey, if you want to get in on the sharing, here's where to do it, and build it out from there.
Yeah, I think that's a great idea. We also talked.
Would it help if I put my email in the chat, and anybody who's interested could just connect?
Would you like us to put that information inside the participants folder for the workshop?
Yes, please. That'd be great.
We can get that taken care of for you, and then I can send out an email when that's added.
We also talked, Cynthia also brought up in our group, and it was really important to perhaps create a document that gives some list of safety things and how to do this, collect this safely, and maybe that's something we could also put in the participant folders, something we could work on together that could go out to parents. And also that explains why they're doing it and why it's important.
Awesome.
There is also a document in your participants folder that has today's registration list. And so if you filled out the pre-registration form that said you were willing to share your information, your email is attached there. So you do have a way to contact other people who participated today who were willing to share their information.
Thanks, Cole. That's awesome. Oh, this is so exciting. I got completely distracted. It's 11:32 and we've got one group that hasn't even said what they've got to say. So let's have group one also share out what they talked about the Denis Sullivan, and I'm just really excited, but I'll stop talking now.
And I will be very brief and to the point, because many of the groups that came earlier definitely echoed our experience. Ginny was very motivational, and if they were taking applications, my name would definitely be on the list, and I would think my group would agree. So I can't wait. And she talked about what kind of mindset you should have if you're part of this experience, and what you might want to expect. So that was really, really neat. And then we also spent a lot of our time talking about the details of the leaf pack and how we might adapt it, how we might use it, trying to kind of tweak and problem solve a little bit. So it was really valuable. It was really neat to have a larger chance for everybody's voice to get heard. So, and if I missed anything in my group, please feel free to jump in. I tried to be brief.
Well, it looks like maybe there's no one else that's jumping in. Anne, did you want to ask your question?
Well, you know, I've been thinking a lot about this. I was really lucky to go on the Denis Sullivan, and I just had 2 questions, and, you know, we're running out of time, but just something to think about. I guess I'd ask, from a programming point of view, it is a very transformative experience. Does that still stay with you over the years? Are you still drawing upon that experience that you had on the Denis Sullivan? And then the second thing is, this is a really different program for Seagull. It's a tall ship. It's not as science, like today was very science focused. You know, we've created a workshop that's not so sciencey, maybe focused more on social studies and shipwrecks and things. And was that as valuable as a strictly science experience? So those are just 2 things I was thinking about. I wish I was in a room with you guys to talk to you about it. Yeah, so if anybody has any thoughts on either of those questions, I'm interested to hear.
And I think, I hope this is okay that I just break in and say this. I would love it if folks could enter that stuff in the chat, because well, I guess it can also be said. I want to make sure that the things you're about to say are captured, and we aren't doing a live transcription, but we are recording. So I guess it is captured either way. Never mind. I retract my statement.
And then honoring people's times. We are over time, so if you do have other obligations, please feel free to leave the session at any point. The rest of us are going to hang in the space for a little bit. If you have questions about either this workshop or the upcoming one that we can answer for you, we'll be here as the facilitators and moderators for a little while longer. And we just really want to thank everyone for participating in our first delve into this realm collectively with Minnesota and Wisconsin teachers, and really couldn't have done it without all of many people's assistance. So thank you, and we look forward to seeing you next week if you do need to leave at this point.